So in the development of the heart, we're going to talk about the formation of the heart fields, the heart tube, the layers of the heart, the looping of the heart tube, and lastly, the different parts of the heart tube. So the most basic thing that you need to know is that the development of the heart is going to take place in the lateral mesoderm, and some cells are going to migrate into its splanchnic layer from the epiblast, and these cells are going to make heart fields. So what are basically heart fields? You can consider them as, basically mesenchyme or mesoderm, because basically everything develops from mesenchyme. Well, not everything. Some things develop from the ectoderm, but some things develop from the endoderm, but most things are from the mesoderm. So there are two heart fields in the development of the heart, the primary heart field and the secondary heart field. The primary heart field develops between days 16 to day 18. Initially, the cardiac progenitor cells are present in the epiblast, beside the cranial end of the perimeter streak. These cells are going to migrate through the perimeter streak to enter the splanchnic layer of the lateral plate mesoderm, and in this layer, they form the primary heart field. On both sides, the cells in the primary heart field become specified from lateral to medial to give rise to specific structures. So from lateral to medial, these are A for atria, LV for left ventricle, and RV is for most of the right ventricle. You can ignore CNT. So now the question is, where does the remaining part of the right ventricle and the outflow tract arise from? The answer is the secondary heart field. The secondary heart field appears between days 20 to day 21, and it's also present in the splanchnic mesoderm in front of the pharynx. And as you already know, it's going to form a small part of the right ventricle and the outflow tract. So now the pharyngeal endoderm beneath the heart fields induces the primary heart field to form myoblasts and blood islands. Eventually, these blood islands will unite and form a horseshoe-shaped cavity, horseshoe-shaped endothelial line tube, which is the heart tube. From your knowledge of the folding of the embryo, you know that initially the heart tube lies ventral to the oropharyngeal membrane, but due to the head fold, the oropharyngeal membrane becomes ventral, and now the heart tube and the pericardial cavity both they lie in the thoracic region. And secondly, due to lateral folding, the caudal ends of the heart tubes fuse, and the central part of the tube expands to form the future outflow tract and the ventricular regions. So just remember that two heart tubes were formed and then they fused together to form a single heart tube. So initially the heart tube is attached to the dorsal side of the pericardial cavity by the dorsal mesocardium. But this mesocardium eventually disappears and the transverse pericardial sinus is formed. The myocardium secretes the cardiac jelly between the myocardium and the endocardium, and this is basically a thick layer of extracellular matrix rich in hyaluronic acid. So now let's come to the layers of the heart tube. The endocardium is derived from the endothelial heart tube itself. The myocardium is derived from the splanchnic mesoderm, and the epicardium is derived from mesothelial cells which migrate from the outer surface of the sinus venosus. So now let's talk about the looping of the heart tube. First of all, the heart tube elongates when cells from the secondary heart field are added to its cranial end. And at the cranial end, you have the outflow tract. So because of this, the outflow tract starts to lengthen, and the tube begins to bend on day 23, and its bending or looping is completed on day 28. Its cephalic part bends ventrally, caudally, and to the right, and its caudal part bends dorsally, cranially, and to the left. So the purpose of all of this is that we need to get the future atria, ventricles, and the outflow tract into their final positions in a fully developed heart. So the red part is the future atria, and they move a little upwards and backwards. And the blue part is the future ventricles, and they move a little downwards and forwards. And the green and blue part is the outflow tract, and this basically forms the ascending aorta and the pulmonary trunk. So lastly, let's talk about these different colored parts. As you know, during this whole looping process, expansions are formed in the heart tube. So remember, this is the heart tube and it just acquired some expansions. So what are these expansions? Let's see. So here's a close-up of the different parts of the heart tube. From bottom to top, you have the sinus venosus, and then the primordial atrium, the primordial ventricle. Between them lies the atrioventricular canal, which is not labeled here. And then after the ventricle, you have the bulbus cordis, and then on top of it, the truncus arteriosus. Between the bulbus cordis and the ventricle, you have the bulboventricular sulcus, which is again not labeled here. So as the name indicates, the sinus venosus forms the venous system of the embryo. The primordial atrium forms both atria. The ventricle part forms the left ventricle and most of the right ventricle. And the bulbus cordis has three parts. Its proximal one-thirds form the trabeculated part of the right ventricle. The conus cordis forms the outflow tracts of the ventricles, and the truncus arteriosus forms the proximal parts of the aorta and pulmonary arteries. The atrioventricular canal connects the common atrium and the ventricle, and the bulboventricular sulcus is going to give rise to the interventricular foramen.